Okay, it looks like our attendee numbers have stabilized, so we're going to get started. Hi, everyone. I want to welcome you. Thank you so much for joining us today for the continuation of our webinar series on hydrobiosis across organisms, scales, and disciplines. For those of you who don't know me, my name is Elena Lazarus. I'm the research coordinator for the Water and Life Interface Institute, also known as WALI, and I'm going to be the moderator for today's talk. It's my pleasure to introduce our speaker today, Dr. Stephen Fried. Stephen earned an undergraduate degrees in chemistry and physics from MIT and then went to Stanford, where he received a PhD studying enzyme catalytics. He then went to the MRC laboratory in molecular biology in Cambridge, where he did a postdoc and made key contributions to synthetic biology and chemical studies. Now, as an assistant professor at Johns Hopkins University, his work on structural proteomics and many more topics, very exciting work we're looking forward to hearing about today, has earned him prestigious honors, including the NIH Director's New Innovator Award. If you have any questions for Stephen during or after his talk, please submit them in the Q&A box at the bottom of the Zoom screen. We'll have about 10 to 15 minutes for discussion and questions at the end. So with that, please welcome Stephen and join me in giving him a round of applause. So Stephen, the room is yours.
Thank you. Thank you so much for the very kind introduction, Helena. I'm gonna share my screen and go into the slideshow mode. Is this looking like everyone can see this in the full screen?
Yes, it looks great.
All right, perfect. Well, I'll thank again for the invitation. I'm really excited to interact with this community. My sort of background training is in protein biophysics and sort of proteomics. So for me to think more about like water and desiccation has been an area that I guess I've been pulled into through, friendships and collaborations. And so it's definitely an area that I think I'm still learning more about, and I'm looking forward to um, input from all of you. So I'll try to make sure I can keep my talk to within the 45 minutes. Okay, so I always like to begin by acknowledging the individuals and organizations without which the work wouldn't be possible. So I'm going to be focusing on actually three projects, which maybe is like one too many, but I just feel like in this sort of audience, there'll be a little bit of interest in all of them. So it'll be a little bit of sort of a sample platter as opposed to going deeply into any one thing. But you'll hear about work from Haley, Egger, and Srimanti in my lab, one project on high pressure biophysics that would not have been possible at all without the key expertise of Richard Gilliland, who is a scientist at the Cornell High Energy Synchrotron. And also, I'll be sharing some work about desiccation that was really led by a star postdoc with Shahar Suganik. This is Sophia Romero Perez, who is at UC Merced, now at Syracuse University. Okay. So to begin with some very sort of basic points. So the Earth can be a very warm, cozy place. But it can also be not. It can be hot, acidic, deep, dark, cold, alkaline, dry, salty. So these all represent different types of chemical and physical extremes that may make ecosystem less habitable to humans and our fellow mesophiles. But of course, the world teems with life that have managed to eke out their existence in basically every possible corner and survive as well as thrive in those environments. And so the question basically in front of us, is what are the sort of the biophysical mechanisms behind the adaptations that are necessary to be able to do well in those types of environments? It probably doesn't require very much motivation to an audience like this, but I just always like to point out that extremophile biology is really interesting. It has sort of implications for astrobiology by sort of teaching us, what are the limits of life and the range of habitability on potential, other worlds. But also it teaches us sort of design principles that can sometimes be applied into protein engineering to design sort of proteins for enhanced stability or tolerance. So there's both an applied angle as well as a sort of out of this world, angle for studying extreme biology. So biochemistry sort of lives through four critical classes of macromolecules: proteins, nucleic acids, sugars, as well as fats. And in order for an organism to be tolerant to a sort of a physical extreme, in principle, all of its molecular components have to be able to tolerate that type of stress because if any one of these four molecules fail, life will probably fail as well because they're all necessary. So, for instance, in the case of lipids, adaptation mechanism can occur very clearly at the chemical level. So, for instance, when we look at um, archaea that, for instance, are adapted to live at very high temperature, we can find, a number of chemical features that make their lipids distinct, such as these these ether linkages replacing the traditional ester linkage between the glycerol and the fatty acids. So these are generally more stable to hydrolysis. And then even some more wonky things such as bilayer spanning lipids that don't sort of have that sort of interstitial area between the two leaflets. So these are very, very different types of molecules that chemically can give thermotolerance. In the case of nucleic acids, it's actually very easy to make nucleic acids thermally tolerant simply by dialing in different amounts of GC content and utilizing certain modifications, such as pseudouridine and to replace uridine. These are really known to stabilize RNA and DNA. And so, actually, if you want to design nucleic acids to be thermotolerant, there's not an enormous amount of sort of intellectual exercise. Like there's relatively simple rules to achieve that. But of course, the topic of the sort of the focus of today's talk is going to be on proteins. I like proteins a lot. I hope you do too. But I think that in addition to liking proteins, I think it's just fair to point out that this sort of they represent the most challenging sort of design problem from the point of view of how to achieve adaptation to physical extremes. And the reason why that is, of course, is because proteins are only marginally stable in the first place. The folded states of most proteins are typically only separated from their unfolded ensembles by just a handful of kcals per mole. Their function requires that they fold into very specific three-dimensional shapes, and typically physical interactions that disrupt those three-dimensional shapes will abrogate their function. And they're also just very highly sensitive to their environment. So this, in many regards, seems like sort of a triple whammy in terms of, why it's going to be hard to be able to engineer or optimize proteins to be able to, stay folded and stay functional under various physical extremes. So to sort of, introduce the method that is really going to be the sort of the star player of today's talk, I'm going to introduce and really talk you through this approach that we did not invent, but which I think we've made some important contributions to, called limited proteolysis mass spectrometry. And this is a method that allows us to, in effect, map structural information about proteins into a format that can be read out on a mass spectrometer, thereby allowing us to probe protein conformation on the scale of entire proteomes. So here's how this method works. It starts with a sort of sample of proteins, which you then treat with this enzyme called proteinase K. So proteinase K is a non-specific protease, which means that it can basically cut between any two residues with pretty much equal efficiency. But it does have a strong preference to cut at regions that are solvent accessible or more unstructured. So in essence, this enzyme allows us to encode structural information about a protein into the form of these cut sites because we expect it to only cut in these solvent accessible locations. We then quench proteinase K after doing just a brief pulse proteolysis because obviously if we let it just go on forever, it'll just cut up proteins to smithereens. We only want it to cut at the most solvent accessible sites. So to do that, we basically add it for a minute and then quench its activity. And then we proceed to treat proteins with the proteomysis favorite enzyme, trypsin, which you can kind of think of as almost being like the foil of proteinase K. in that it's very, very sequence specific. It only cuts after arginine and lysine, but it's very insensitive to structure, meaning that it's very good at accessing those arginines and lysines regardless of how buried or exposed they are. So what is the result of this double digest of, treating with proteinase K and trypsin? Well, on one hand, you're going to expect to find so-called tryptic peptides, which are those where both the ends of the peptide can be justified from a place where trypsin made a cut. But we're also going to find peptides such as the one here that I'm calling a half tryptic peptide, meaning that one of the cut sites came from trypsin, but the other cut came from proteinase K. And of course, the reason why you can know that is because once you've sequenced this peptide on a mass spectrometer and you align it back to the reference proteome, what you would find is that there is not an arginine or a lysine in the position that directly preceded it. So that would imply that this cut could not have come from trypsin. It had to have come from proteinase K. And what that means is that this residue had to have been solvent accessible in the protein that this particular peptide came from. So this kind of gives you an example of what those like sequence rules look like, where this is, for instance, a peptide that we sequence, and we know that this cut between Q and T came from proteinase K, because that's not a place where trypsin would be allowed to cut. And that would mean that Q31, for instance, was solvent accessible. So because in a typical mass spec experiment, we can normally sequence between, let's say, 20 and 50,000 peptides in one experiment, we can basically get a structural snapshot of an entire proteome, which is really powerful. So one sort of the thing that we've been using this method for traditionally is for looking at protein refolding. So what this experiment basically does is that it takes this sort of limited proteolysis workflow that I told you about, and it couples it to these global unfolding refolding reactions where we take cells, we break them open, we get their extract out, we take that sort of complex soup of proteins and subject it to global unfolding, where we basically unfold all the proteins by adding high concentrations of denaturant directly to the extract, and then in essence challenge the proteins to put themselves back together again by diluting the denaturant out and seeing which ones can resume the form of their native fold versus which ones get lost along the way and essentially adopt some incorrect misfolded form. And of course, to then figure out which proteins were the ones that folded correctly versus the ones that couldn't, we then take both samples, the native, which we're using as a reference, and this refolded reaction, which we're using as our test, subjecting them to this limited proteolysis. And the notion is that if a protein is good at refolding, it really would have no memory of the fact that it was ever unfolded. So we'd expect the exact same sort of profile of peptide fragments to be the same in the before and after. So you can see that, these are sort of the same, whereas this protein that failed to refold, we would basically see unique fragments appear that correspond to locations in the protein that only became solvent accessible upon the protein failing to refold. So this is sort of, an initial application that our lab utilized this LIP-MS for. But what I want to sort of stress for now is that, refolding is really only, one type of stress that we could imagine. There are a lot of other stresses that we could imagine applying to proteins and then using this same type of experimental technique as the readout. And the first, I guess, like scientific sort of story that I want to focus on is looking at the effect of high pressure. So, of course, the reason why we're interested in high pressure is because it's the prevailing physical status of being deep in the ocean. So the ocean floor is on average cold at about 3 degrees Celsius, high pressure with a sort of like typical pressure of around 30 megapascals, corresponding to a depth of about 3,000 meters. That's the sort of the average depth of the ocean. Of course, there are many places along the ocean that are even deeper than that. The trenches, like the famous Mariana Trench, extends 10,000 meters below the surface. So that has a pressure of about 100 megapascals. So that's about 1,000 times higher than the ambient pressure on the surface. And yet, despite that, these like deep sort of locales within the ocean are teeming with life, and actually not just bacteria, but even fish and jellies and complex eukaryotes. have all been able to figure out ways to adapt to these exceptionally crushing pressures. And so the question that we basically want to know is how do sort of everyday proteins, like the ones that would, be in organisms that are surface dwellers, how would they perform under high pressure? And so here's in essence the experiment that we're going to do to test that. So we're going to take a surface dwelling organism. In this case, it's the bacterium called Thermus thermophilus. It actually does like to live at high temperature, but it does not like to live at high pressure. It only lives in sort of surface springs, surface hot springs. We're going to take this organism, we're going to lyse it to get the proteins out. We're going to take that extract and do one of two things to it. We're either going to subject it to limited proteolysis, this sort of method that I told you about, or we're going to take that whole extract and subject it to very high pressures and then perform the limited proteolysis under high pressure. And so the notion is that if a protein is sensitive to pressure, then we might expect there to be regions of the protein that might become deformed under that sort of perturbation, which would then allow proteinase K to cut in a new location, which would then which would then cause one of the tryptic peptides to get sort of split up into these smaller half tryptic peptides. And then we can sequence those things, see where they lie within the protein, and learn about what regions within proteins are more sensitive to high pressure. So it's worth, I think, pointing out that this required a sort of, Rube Goldberg, like instrument to be able to perform this like mixing at ultra high pressures. And none of this would have been possible without sort of like a really lovely collaboration between my team and Richard Gilliland, who is a high pressure expert at chess and has spent most of his career doing high pressure sacks. And then when we sort of, first met at a NSF Research Coordination Network event, we got excited about the idea of sort of taking high pressure biophysics to the proteome sort of stage. Okay, so what do the data look like from an experiment like this? So one of the ways that we can represent the data from these experiments are these like volcano plots. So what these volcano plots represent is every single dot that you see corresponds to an individual peptide that we are able to confidently sequence and quantify. And in this case, we're looking at the relative abundance of that peptide in these pressurized samples relative to ambient. The x-axis is an effect size, so it's how much more abundant that peptide was in one sample versus the other. And the y-axis is a statistical measurement of how confident we are that that difference is real, that it's not just sort of within the error or the noise. And so you can see that there are a lot of sort of like, dots that are sort of like toward the middle. They're actually kind of all on top of each other. So it's kind of easier to see how many there are on the histogram. So these are basically correspond to regions within proteins that are not responding to the pressure treatment, meaning that they have the same abundance in the before and after, meaning that they have the same susceptibility to proteinase K in the before and after. But then you also see this very large cluster of dots far off to the right. And what these correspond to are peptides that we effectively could only identify in the sample that was highly pressurized, and they were absent in the sort of ambient samples. So what this tells us is that when you take a proteome and you subject it to pressure, there are wide scale changes in structure that then in turn cause the proteome to become more susceptible to proteolysis. And because they're now more susceptible to proteolysis, we generate all of these new fragments that we can start sequencing, which were not present in the native sample because these represented regions that were well packed enough in the protein that they were not targets of proteinase K and therefore could not generate those peptides under ambient condition. So when we map those peptides, we basically make the assessment that if you go to these pressures, very, very high associated with the Mariana Trench, 100 megapascals, we find that about 40% of the proteome is structurally perturbed, and actually about 20% of all peptides that we were able to sequence were distinct. So it actually, though, was not nearly as significant of an effect at that 50 megapascal, so half the pressure. In that case, actually only 12% of the proteome was structurally perturbed. So it seems that um, there's sort of a good bulk of proteins that would not be affected by high pressure unless you go up to 100 megapascals, but at 50 megapascals is more of a specific subset. So that of course invites the question, well, what types of, features or what types of I guess traits about a protein would correlate with its ability to withstand high pressure? And this is where, in my lab we have a lot of fun. Once we've, done these, hard, tricky experiments, we get these sort of big data sets with, a few thousand examples, and it allows us to have enough information to try to look for trends or patterns. And here's what we found. What we found is that proteins that are resistant to pressure tend to have cofactors associated with them with higher frequency. They tend to be closer to neutrally charged. So that means that if you look at their sort of distribution of isoelectric effect, what you find is that things that have an isoelectric effect near neutral tend to have a lower rate of being perturbed, whereas things at the edges of the isoelectric distribution, so things that are very acidic or very basic, are more likely to be perturbed. But actually, the trend that I want to focus the most on is this trend in relation to packing density. So what we're seeing is that if proteins are more tightly packed, they tend to be more perturbed, whereas if they are more loosely packed, they tend to be less perturbed. So what that means is that proteins that are resistant to pressure tend to have sort of like extra space. So how should I think about packing density? What actually does this term mean? So the sort of like familiar sort of mathematical definition is that we all have like seen that if you have like a stack of oranges at a grocery store, the oranges can't completely fill up the space because spheres basically don't tile Euclidean space with perfect efficiency. In fact, only about 74% or pi divided by 3 times the square root of 2 of the space is occupied when you sort of try to densely pack spheres. So that means that the packing density of spheres is 74%. Now, we can also calculate the packing density of proteins. They have sort of more idiosyncratic shapes compared to spheres. But because of the, advances in artificial intelligence-based protein structure prediction, we can basically, go to the AlphaFold database, start with, the 2,000 different Thermophilus proteins, say, hey, this is a pretty good, model of what the structure looks like. Take those structural models from AlphaFold, hydrogenate them, because obviously AlphaFold structures don't contain protons, but protons still do take up some volume. And then take those protonated structures and subject them to a calculation using this algorithm called protein volume that basically tries to figure out like how much extra space is there within the internal interstitia of a protein. And you can see that there is variation, although the variation actually is within like a somewhat narrow range. So pretty much all proteins have a packing density between 70 and 80%, with the median being close to 74%. And so what basically Haley and Richard discovered is that if you're on that sort of, more densely packed, side of the distribution, so packing densities that are closer to 78%, those are the ones that seem to get perturbed by structure to a greater degree, whereas the ones that are on the looser and, with packing densities closer to 70%, are affected by have perturbations occurring less frequently. And this actually, if you sort of like stop and think about it, makes a lot of sense, right? Because basically what happens when you take a protein and you put it under pressure, well, when you put anything under pressure, you're basically going to subject it to a compression. And in essence, if a protein is already very, very densely packed, then it's not really going to be able to compress any further without starting to interpenetrate the van der Waals radii of the atoms. That, of course, is going to have a very steep energy penalty because as you look at the sort of the van der Waals potential, as things start to get, too close to each other, the energy shoots up very fast. So it ends up being um, it ends up being costing less energy for simply the whole chain to unfold. And then that represents a solution that doesn't require sort of van der Waals sphere interpenetration. On the other hand, if the structure of the protein is just kind of like looser, and there's a little bit more give, there's a little bit more sort of leeway, and that means that under high pressure, you can then imagine that this allows the molecule to compress in a way that does not cost nearly as much energy and can accommodate that sort of perturbation without causing sort of steric repulsion. And it's worth pointing out this had actually been noticed in a very early study by X-ray crystallography, in which a piezophilic protein, in this case, it was this sort of specific dehydrogenase, this 3-isopropyl dehydrogenase. It had been shown that it had more voids when its X-ray structure was solved in the early 2000s. So in essence, the idea that we're putting forward is not entirely new, but we are now able to say that this is true on the proteome scale and not necessarily just the case for this one enzyme with an X-ray structure solved. So I think this kind of highlights the power of these sort of proteome-wide approaches, computational approaches to be able to generalize knowledge and extract sort of like, unifying physical principles. Okay, so that was the first vignette. Let's move on to a story about desiccation, which I know is something that a lot of people in this room know a lot more than me on it. So desiccation is the loss of nearly all the water within a particular system. It is a stress that actually is quite common on Earth and probably is going to become more common in light of climate change. And but like the ocean floor, these very dry environments like deserts are still teeming with life, again, from all kingdoms of life, not just bacteria. But the key sort of point here is that the loss of water actually represents an enormous problem for proteins because proteins, of course, sort of have a very sort of complex surface structure, and those surfaces are constantly interacting with water in the form of hydrogen bonding and other sort of electrostatic solvation interactions. And so the removal of water basically causes the loss of a set of interactions that play a key role in stabilizing proteins. And so the question is there a way to sort of accommodate that and to sort of keep proteins folded and keep them stable even once these sort of key surface interactions have been abrogated by the total loss of water, such as during desiccation. So these are experiments that, as I alluded to during the introduction, were led by Shachar Stikhanek's team and his talented postdoc, Sophia Romero Perez. Here's the experiment. Basically, what we're going to do is take, in this case, yeast, dry it out fully in a desiccator, and then, or sorry, first take yeast, first lyse the cells to get the extract out. Take that protein extract, totally dry it in a desiccator, and then rehydrate it. And there's basically actually two questions that we're going to be interested in. The first is we're going to be measuring a property called resolubility, which is to say, once I add the water back, for each individual protein in yeast, what percent of that protein can get resolubilized, can basically be re-entered back into the soluble phase. So to put like a number on that, like if a protein is, 100% resoluble, it means that once you add the water back, 100% of the protein goes back into solution. Obviously, 0% resolubility would mean that it all basically fails to get rehydrated. And in addition to measuring resolubility, this can be sort of measured through quantitative proteomics by doing pelleting to basically separate the soluble from the insoluble fraction. Another measurement that we're going to do is our favorite LipMS experiment. So this is essentially measuring structural retention. So meaning that there is ways that proteins could maybe get resolubilized, but still get resolubilized in the incorrect conformation, right? So it's possible that protein A, you dry it out, you add water, it goes back into solution, but it goes back into solution and now has a sort of deformed or altered sort of structure, which is an irreversible sort of perturbation from its original native form. So in this study, we actually measured both the resolubility as well as this structure retention property, which is what we have access to through LipMS. Okay, so what do the data look like? So first of all, we find that in yeast, most proteins are not very resoluble. So the average protein in yeast, only 25% of it can find its way back into solution. So this is actually not something that sort of like the prototypical yeast protein seems to be all that good at. Now, what's interesting though is that if you sort of then sort of separate, the proteins that have their structures retained according to LipMS versus having their structures perturbed, what we find is that the proteins that have their structures retained actually tend to have a bump up in their resolubility. So that shifts that distribution closer to about like 40 or 50%. You can really see it quite clearly here that when we look at proteins that are the least resoluble, so we'll call that this kind of like lowest resolubility quantile. They very, very rarely retain their structure, whereas proteins that are good at getting resolubilized, they do retain their structure. So that kind of actually gives us maybe a little bit of like a mechanistic thought that maybe what's going on is that the proteins that are resoluble are effectively ones that can retain their structure. And because they can retain their structure, that's what then allows them to then basically, remain in this sort of native soluble format. Whereas the ones that basically crash and burn out of the solution are the ones that basically lose their structure. And then that is that perturbed structure that then is what actually makes them insoluble. So again, we have a large data set. So that means we can start to play with, well, what do things have in common? This is a bit of a complicated figure, but I'm just going to try to like walk you through it relatively quickly. There's really, I would say, kind of like two things that I want to draw your attention to, and that is the green and the red. So what is going on in the green is that it's telling us that proteins that are very good at getting resolubilized, so that means that they're in this like highest quantile of resolubility, they tend to be very abundant. They tend to be some of the more abundant proteins in yeast. And that you can kind of see that like as you go to the lower resolubility quantiles, that sort of median abundance tends to drop down. But actually, I would say probably the single biggest sort of determinant that we found in this data set is that if you look at the most resoluble proteins, what you find is that they have actually very little disorder. So they tend to be highly folded or highly globular proteins. And actually, the more disorder you add into a protein, the more it seems to sneak into these lower resolubility quantiles. So that's actually a little bit counterintuitive because I think many of us, are very aware of these, like for instance, cause proteins and tardigrades, which are highly disordered proteins that promote sort of resolubility. But that might be an exception. Cause are special in that sense. It seems that for your run-of-the-mill protein, your disordered regions are actually a bit of a liability in terms of getting things back in system, which maybe also makes sense because disordered regions are sort of able to like, talk to and interact with a lot of other things. And maybe that basically creates a bit of a problem in terms of sort of keeping things nicely packed and tucked away in the dry state to make them ready to get resolubilized. So, okay, so that seems like that was like, kind of like an interesting start, but can we go further? Can we actually like look at these like sequences, look at these structures and really, try to come up to it with a stronger sort of, sense of what do now that we've kind of, been able to say, okay, like, disorder is not good, like it's better to be globular, but then it's not like all globular proteins have the same resolubility. So what separates, the best from the best of the best, right? If we were to look at, these domains versus these domains, which types of domains seem to, be better at getting rehydrated? And so here was the analysis that we did. This was led by Alex Holhouse's team at Washington University in St. Louis. They basically take the protein structure predictions from the AlphaFold database, lop out the individual domains from those proteins, and then they use an algorithm that identifies which of the residues on those domains are on the surface. And then we simply do a counting exercise. We just count how many residues of each residue type are on the surface of the globular domains. And what this sort of data table is showing you here is that there's actually very, very strong enrichment patterns for the choice of residues that you have on the surface of your globular domains. Specifically, proteins that have really crappy resolubility tend to have a large number of positive residues and a large number of polar residues on their surfaces. Whereas on the flip side, proteins that are in the higher resolubility quantiles tend to have a lot of negatively charged residues and a lot of what we're calling small residues, which are the ones that have these like relatively small side chains like A, G, and P. And this is actually a pretty pronounced effect. To sort of like, put that sort of in context, if we look at like structural perturbation, what you see is that if you have a protein that has the low end of the amount of acidic residues on their surface, 15% of those peptides get perturbed when we do this rehydration cycle. Whereas if we look at the opposite extreme, of proteins that have the highest number of negative residues on their surface, only about like 5% of their peptides are getting perturbed. So there's about a threefold effect there. And that's a threefold effect over tens of thousands of different sites. So like the p-value on this is like 10 to the minus 200. It's a very significant effect. And you see that sort of same sort of strong effect with those like small residues. So A, G, and P, we find that these are very enriched if we're looking at the proteins that have these are very enriched, I would say. Sorry, let me say it in a different way. If you have a large number of A, G, and Ps on your surface, then you don't have very many perturbations. But if you lack A, G, and P on the surface, then you tend to have more structural changes upon desiccation rehydration. So in essence, there seems to be like a specific set of residues that are statistically enriched. The next question that I think you want to ask yourself is, okay, you've discovered some pattern, but is it just statistics, sort of is it just kind of like the classic problem with like big data, where the more data you have, the easier it is to find a correlation? Like, can you take that prediction and really put it to the test with an orthogonal assay? And this was, I think, a really beautiful experiment that Sophia did that did exactly that. She took, the green fluorescent protein, which natively is actually not a very desiccation resistant protein at all. And she put a lot of negative charges on the surface of the protein and discovered that indeed, just making the surface negatively charged gave this protein the ability to quantitatively rehydrate upon desiccation. So it does seem that this is a principle that can be used for engineering, and we're interested in seeing in the future if this can be exploited in the context of climate mitigation measures and trying to sort of make organisms more resilient to sort of extreme dryness events. All right. So I'm going to try in the last 5 or 6 minutes, maybe it'll be closer to 7 or 8, to tell you a little bit about our work on thermophiles. So I'll sort of like preface by saying that actually we've been working on thermophiles for longer than any of these other projects. And actually it was the sort of like the OG extremophile that got me initially interested in this sort of area of research. But it's also the data, it's also the experiment that has given us the most confusing data, and where we've had to basically sort of revise our model many, many times to try to kind of come to an understanding of what's going on with it. So it's still actually kind of a work in progress, but I think in the last, I would say, few months, we've come across some, new results that are giving us more confidence that we think we understand what's going on. And so, to sort of like introduce this topic, our original experiment to look on thermophiles was this refolding experiment. So again, remember LipMS, it basically tells you, is the protein structure the same in the before and after? We've looked at the before and after being desiccation rehydration. We've looked at the before and after being, pressurization versus non-pressurization. Now we're basically going to go back actually to the original experiment that my lab did, which was looking at refolding, except now we're going to look at a thermophile. We're going to take the proteome of thermus, unfold it, refold it, see if the proteins can put themselves back together again. And the answer is thermus proteins are pretty bad at refolding. They're actually the worst of any organism that we've seen before. And that really surprised us, right? We were expecting, oh, it's a thermophile. These proteins are rocks. They have to be good at refolding. And the result was the exact opposite of that. And so you can imagine that sort of made us scratch our heads. We repeated the experiment a bunch of times, got the same result. We also wondered maybe there's just something sort of specific about the phylum that thermus lives in. So Thermus happens to live in a phylum called Dinocochota, which also contains Dinococcus radiodurans, which is not a thermophile. And if we perform our same unfolding and refolding reaction on Dinococcus radiodurans, we find that actually it refolds very well. So in essence, there's nothing specific about this phylum that makes it bad at refolding, but there is something specific about this thermophile that makes it bad at refolding. So how can you explain that? So the model that we were sort of kicking around for the last 2 years was that we sort of like drew some like very basic looking sort of energy landscapes. And like, this is sort of like the typical, model of, protein folding with these like funnel shaped energy landscapes in which the native state represents some global thermodynamic minimum. And I think it's fair to say that when we sort of think about the textbook picture of what is going on in a thermophilic protein, we tend to imagine that their native states are even more stable. So they have these deep sort of wells that in essence sort of keep their native states folded because of this very large free energy of unfolding. But it's actually possible to imagine an alternative where the main sort of biophysical feature that supports thermophily is actually having high barriers. So meaning a high delta G double dagger as opposed to a high delta G of unfolding. So in this sort of model, the thermophilic protein is actually not any more thermodynamically stable than the so-called mesophilic equivalent, but it just has a very high barrier to get out. And the reason why that sort of model seemed appealing to us is because we figured that this could be consistent with our refolding data, right? That in essence, if the structure of the energy landscape looks like this, then the protein is going to stay folded at high temperature because of those high barriers. But now, if you do force that protein out of that well, it's now going to have a super hard time folding back to its native state, in essence, because the barriers are going to work in both directions. So is that true? Well, this is a complicated slide that I'm not really going to go into in any serious detail, but I'll just sort of say that we did kind of classic protein sort of biophysical characterization on two purified proteins from Thermus, namely PGK, phosphoglycerate kinase, and GMK, which is guanylate kinase. And the kind of key result here is that these are proteins that are extremely slow to unfold. If you basically unfold them at high denaturant and high temperature, they take on the order of hundreds of minutes to unfold. So they are very kinetically stable. But the sort of the key result here is that if we actually look at their thermodynamic stability, they're actually quite unexceptional, meaning that like in sort of conventional urea melts, we find that they have midpoint concentrations that are actually not that high. Now you might be wondering, how come like people haven't known this like since like forever? And the answer to that question is in order to do these experiments, we did something that people do not conventionally do, which is each one of these points represents an individual sample of protein that we put in denaturant and we incubated it in denaturant for 2 weeks before taking the CD spectrum. So in essence, basically saying, let's actually let these proteins really have enough time to equilibrate. And if you give thermophilic proteins enough time to equilibrate, what you find is they're actually not that thermodynamically stable. But what really is the case is they just take a very long time to unfold. And so what that suggests is that maybe the sort of the key sort of factor in thermophily is kinetic stability rather than thermodynamic stability. At least we have direct evidence for that for two specific examples, and that seems to also be consistent with the proteome-wide result of low refolding capacity. So I'm going to maybe just like skip this last bit because I want to make sure there's time for questions, but I'll just sort of say that we've been sort of curious in the most recent sort of months to try to do some structural informatics to look at some of the structural features that support this sort of like high kinetic stability, and I can talk about that if there's interest in the Q&A, but if there's more interest in the Q&A about desiccation or high pressure, then we can go back to that. So I'll just conclude by saying that the proteome can adapt to many physical extremes and that those adaptations can occur through subtle alterations in protein sequence and structure. We think that adaptation to high pressure is possible by sort of carving out tiny little voids within the structure of a protein that can act as little cushions against compression. We think that adaptation to desiccation is possible through the sort of like, choice of by a judicious choice of amino acids on the surface of the globular domains and by sort of limiting the amount of disorder. And we think that kinetic stability plays sort of like the key sort of role in allowing proteins to stay folded at a high temperature. And with that, I'll stop and take your questions.
Thank you so much for the amazing talk. It's really inspirational to see those applications and the different areas and the techniques your lab's been working on. We've got a couple questions that are already starting to pop up in the chat. Please feel free to use the Q&A button at the bottom of your Zoom screen to enter your questions. If you want to like voice your question, ask it yourself, you can raise your hand and I'll unmute you, but we are going to go in order. We've already got a couple questions rolling in. Um, I also wanted to do two quick reminders before we start questions that this is our first talk of 2025, but we are continuing the seminar series first Thursday of every month. So if you enjoyed this talk, we hope to see you back for future talks. You can follow us on social media or visit our website, and registration is already open for those later talks. So, with that, we'll start with the first question in the chat from Charles. Regarding your desiccation experiments, did you prep the yeast for desiccation via starvation or osmotic stress before lysis and proteomic studies? They noted that the proteome will change during the transition to dormancy.
Okay, so this is a great question, Charles. Yeah, thanks for asking it. So for this like initial study, we were intentionally trying to like make it so that like proteins would fail, right? Because if every protein could rehydrate, then we wouldn't really see, the differences that we were hoping to be able to, kind of like, categorize things into different groups. So in essence, the answer to your question is these were yeasts that were grown to log phase, which, as you point out, have not really been sort of like adapted to sort of stress, which would happen if you were to grow them to saturation. Then various sort of circuits kind of kick in, and yeast tend to produce large amounts of trehalose. So we absolutely expect that if you had grown the yeast to saturation, they would have produced trehalose, and then when we would do these desiccation cycles, that would then have sort of given an additional sort of resource to help the rehydration. In essence, what you're kind of seeing in this experiment is what we might call like the intrinsic, rehydratability of each protein. And I think that's like an important point to point out that like when we think about desiccation intolerance, at least like, in sort of like our collaboration, we tend to sort of think about it as having these two sort of like very sort of separate, elements to it. On one hand, you have sort of desiccation resistance that comes in the form of producing osmolytes, which requires having modules that can sense the environment and then activate certain circuits, then overproduce those certain kind of key small molecules. And then you also have the so-called intrinsic I guess mechanisms of protection, such as having these like specific types of residues on your surfaces. And I think that both are important. It's probably worth pointing out that a yeast relies actually more on the first rather than the second, as evidenced by the fact that its inherent, sort of resolubility is actually really bad. But then what's interesting is organisms like Deinococcus radiodurans might be more on the side of, intrinsic, resistance. And we know that because in separate experiments I didn't tell you about, the rehydratability of the Deinococcus proteome is actually really high, even without any priming. Um, and that's interesting, right? Because, these like sort of intrinsic mechanisms of resistance, like in theory, might be more helpful in like, times of like acute desiccation where it occurs so quickly that, cells wouldn't necessarily have enough time to like sense and react. So I think that like, both of these are sort of worth talking about, but you're absolutely right that the way we designed the experiment, purposely made things hard on yeast and purposefully were kind of designed to sort of focus on these like intrinsic mechanisms as opposed to the sort of osmolyte based, sort of routes.
Okay, great. Another question in the chat. Do you think there would be a structural evolutionary fingerprint differentiating between kinetic and thermodynamic stability?
Okay. Yeah. So this is a really good question, one that we've definitely been thinking a lot about. I think that, and it's worth like pointing out that like what we're saying is like kind of weird, right? Because like we know that you can create like highly thermodynamically stable proteins, right? Like just ask anyone, in the Baker lab and they'll like whip out, like a protein that, has like ridiculously high stability. And we've actually done a few sort of, experiments ourselves to like double check that, they're not wrong, right? Like if you do long incubations, long equilibrations of, de novo proteins and then measure, equilibrium stability under the conditions of long equilibration, they really are, in fact, thermodynamically stable. And it's just, that does not seem to be the route that was taken by actual extant proteins in extremophiles. So then the question is why? And this is like a preliminary answer, but I think the reason why is that I think there's basically two reasons. I think one is that it's just easier. I think that it might just be that it takes like a more specific set of like sequence considerations to achieve thermodynamic stability. And it might just take fewer adaptations to achieve kinetic stability. And it just ends up being kind of like, the evolutionary path of less resistance. That like if I can get away just making, five mutations instead of 20, then and that basically gets things to be good enough, then that's basically the path that evolution can take. But it is actually also possible to consider that there might be more of, a adaptive function to this, right? That proteins that are thermodynamically stable, maybe that represents a scenario where then there's more trade-offs with them not being very dynamic or being very flexible. We know that, sort of de novo design proteins tend to be, bad at, sort of, performing functions that require multiple conformations. So it might be that kinetic stability sort of is the solution that basically allows you to have the best of both worlds, i.e., stay folded at high temperature, but not at the loss of like losing dynamics and sort of like, interesting sort of, multiple conformations at the well bottom. So again, speculative, but that would be sort of at least a hypothesis.
Great, thanks so much. We have two questions about some of the high pressure aspects of the limited proteolysis. So Olivia was wondering, do the high pressure experience not affect the activity? And also, Peter was wondering if they're resistant to the pressure. Kind of how do you validate the unimpaired activity and specificity of these under pressure? Yeah.
Okay, great. So let me answer Olivia's question. So we did do some separate control experiments to ask the question, does the high pressure impact the actual activity of proteinase K itself? Which if it does, that would be a very significant, sort of um, what's the word? Problem or I guess like sort of like confounding variable for the analysis that we have, and it's actually especially sort of um important because there has been research that has shown that pressure can of course influence enzyme activity actually in both directions. Like sometimes and sometimes high pressure can activate an enzyme, and the sort of the theoretical framework that's used for that is like the delta delta V double dagger, so that essentially if the transition state involves compression of the reaction coordinate, that actually pressure would be expected to sort of activate that type of reaction. And there are some enzymes where pressures such as the ones that we've used have been reported to activate by as much as, like 5 to 10-fold. So the experiment that we did is that we basically did a simple fluorescence assay where we had like a peptide with a fluorophore and a quencher, put that inside, our high pressure apparatus, did the limited proteolysis with proteinase K, and then measure this sort of activity of the enzyme as read out by essentially trying to like decouple, activity changes in the enzyme with, pressure-induced structural changes, because this little reporter peptide shouldn't undergo any structural change. So therefore, we're kind of isolating the effect of pressure on the enzyme. And actually, we were just really lucky and there was no detectable difference. So it just happens to be that proteinase K has a transition state where there's not a very big delta V double dagger. And that was just dumb luck. It could have been that, we did that control and it would have like destroyed our study. which would have been very sad. But it happens at proteinase K. And then you also ask about trypsin. We don't worry about trypsin actually because trypsin is not even done at high pressure. So really the sort of the only step that is really sensing sort of conformation is just the proteinase K step. And then trypsin, yeah, we just do that actually under ambient pressure anyway. And then the last question by Peter, as I understood, lip was done under the high pressure is dependent on are they resistant? Yeah, so proteinase K, yeah, as far as we can tell, the activity was insensitive to pressure based on this like little fluorescence assay. And I think that's like figure S3 in the paper, but it's not in my slides.
Great. And then there was a second part of Olivia's question. Looks like she's wondering if you repeated the or have considered repeating the experiments with species isolated from the Marina Trench.
So what we want to do for like the next chapter in this sort of line of work is to, as you're basically alluding to, perform these experiments on a piezo file. And I think it's abs and actually this is actually I'm really glad that you asked this question because I need to make actually a very important like caveat to what I claimed which is that so I showed you that for thermos, which is a piezo-sensitive organism, that the proteins that are the more resistant to high pressure are the ones that have like lower packing density. So then maybe you would expect, well, maybe then if he were to take the alpha fold structures of a piezophile and computationally, calculate the packing density of all of the proteins in a piezophile, based on what I've told you, maybe you would expect that they would have packing densities, that are shifted to lower numbers. So the distribution of the packing density of a piezo file would be, shifted in, to lower numbers compared to a non-piezo file. And actually, that does not seem to be the case. We've done that. So what we can basically conclude at this point is that amongst non-piezo files, having more voids seems to be a protective mechanism, but that does not necessarily seem to be the mechanism that was exploited by piezophiles. At least we don't have evidence that supports that yet. So we really, actually, I would say like we need to do these experiments on piezophiles because we just don't know yet what's going to happen. Like there might be other operative mechanisms within that context. In terms of like Mariana Trench, the organism that we were thinking of working with first is this bacterium called Photobacterium profundum, which is a organism that lives normally. I think it lives around like 3,000 meters. And the reason why we picked that one is because it's a facultative piezophile, which means it can also actually grow at ambient. Most things that grow in like these ultra, deep places they're obligatorily piezophilic, meaning that like they just die if you bring them to the surface. And the reason why that's a problem for us is because when we like prepare our samples, like even though we do the limited proteolysis at high pressure, we still we still have to lyse the cells and sort of clarify the lysate and prepare the sample at room temperature at room pressure before we repressurize it to do our assay. And so if you had something that like lived in Mariana, like for instance Colwellia, those are these really really piezophilic organisms. I think we would be worried that by the time you basically have the cells and you lyse them and you basically get the experiment ready, are you now no longer looking at a native proteome anymore? So in essence, that experiment would require what's known in the field as isobaric transfer. You'd have to basically culture the cells at high pressure and then basically prepare the sample from the cells and load it into our device all at pressure. And we don't yet have the tool to do that. But I think in some ways that would actually be a really cool experiment because then you can almost do the opposite experiment, right? Like basically grow up a piezophile, isobarically transfer it into the apparatus, do lip, and then now that's like your reference condition. And then your test condition is to then bring it back up to the surface and then see what changes, right? So that would be a super interesting experiment, be almost like the inverse of what we've done, but it would just require sort of the ability to move the sample without depressurizing it, which we don't yet have.
Well, very cool to think about. Thanks so much for those thoughtful answers and for the thoughtful questions from the audience. I think we've answered everything in the Q&A chat and we're at time. So I believe we're going to roll out for the day. I just want to give a round of applause and thank you again so much for the amazing presentation and the wonderful discussion. I'm sure if anybody has follow-up questions, they can reach out. And we'll be back next month for another talk. Hope to see all of our audience members back again. So thanks so much. Have a good rest of your day.